Hi everyone, I wanted to take a moment and talk about the shapes of bacteria, also known as bacterial morphology. Morphology is just a fancy word for shape, as in I'm looking at particularly how the bacteria is formed, and that relates to its function, its class, and its family. The first shape that we see here is an oval shape, a circular shape, uh, very beady looking thing, and that would be cocci. A singular bacteria in this shape would be coccus. Something that would be shaped like this would be something like streptococcus, which is strep throat, or staphylococcus, which is staph infections that you might find in hospitals or veterinary offices. The one in the center here, bacilli, is kind of more of a rod shape. It's a very skinny looking one. And sometimes they will have little flagella off the backside there to use as little things to maneuver around. A good example of this would be, oh gosh, Bacilli anthracis, which is the bacteria that causes anthrax, that disease that everyone was worried about being mailed from one place to another back in the early 2000s. Um, let's see here. And then there's Spirilla, which, again, Spirillus is the singular. In this case, it's just a kind of a curvy structure that's in a spiral. They will have some kind of flagellum to maneuver. They're actually spinning around like a top to be able to use for locomotion. A good one as an example for this would be leptospirosis, which would be whale's disease. It's a disease that you get if you drink infected water that has been infected with, like, say, deer urine or dog urine or wolf urine or something from the wild there. Not a very common disease for sure, but it is something that is bacterial and can really hurt you and mess you up. Now, in addition to talking about how these individual bacteria look, we can take a look at how they're actually constructed in terms of colonies, because these arrangements are important for when we're looking at individual colonies to see exactly what is more likely for them to be. For example, Streptococcus is more likely to be individual colonies like a diplococci here, whereas Staphylococci is going to be swarming with a bunch of different ones. Tetracocci is going to be just four of them. A chain of cocci is likely something related to pneumonia and stuff like that. Each bacteria has their own way of arranging their colonies because they have different ways of communicating with one another. So this is a good way to understand not only what the bacteria look like, but how they're arranging can be a way to we identify them. Bacilli bacteria will either be individual bacillus or a chain of bacilli. Uh, we actually see this with some staph infections as well. And the spirilla bacteria will be individual more often than not. You won't see them in colonies necessarily because they have ways of maneuvering around. These individuals are often in colonies. They don't have as many ways of maneuvering, so their only way of going from one place to another is reproducing, really. Like this individual cocci reproduces into two. Well, now it's in a different spot than it was before because the other one takes up some space as well. Not to say that they don't have appendages of some kind. You'll see that in medical interventions when we study bacteria, but really there's not much movement going on with cocci or bacilli compared to spirilla. They move, not so much as always, though. Now, in our next activity, we're actually going to be looking at identifying bacteria a little bit further because some look pretty identical under the naked eye. But we have a way of actually testing and treating an identity to try to separate these bacteria into classes called gram-positive and gram-negative. You can kind of tell that the test that we would do is called a gram test, or a gram staining as we call it. Because what we're going to do is we're going to stain them with both of these colors. And depending on the structure of the bacteria, one color will pop up. Either this purpley color if it is a positive for a gram test, or a pink color if it is negative for a gram test. Now, what exactly do I mean by structures? Well, want you to take a look first at the gram-negative one. This one would be pink after a gram staining. You can tell that the structure of the cell wall here is much different. It has a bunch of what are called lipopolysaccharides, which are just sugars that have some fats on the end of them, and it has an outer membrane made of phospholipids. We've heard of those things before. And it only has one of these strong and rigid, what are called peptidoglycans. Peptido meaning protein, and glycan meaning of sugar. So these are sugar protein connections over and over again, making a very strong wall between the inside of the cell and the outside of the cell. A gram-negative one is pink because it only has one of these particular walls, whereas a gram-positive will turn purple because layers and layers of those stains are coming together. And therefore, if I have all these all stained, then the thickness will make it look a little bit darker compared to this light color of pink. It'll be more of a purpley pink color. And that is why this is such an effective test, because we can literally see the differences in the structure of these two bacteria and really compare them in terms of class then. By the way, just as an aside, you might suspect that the gram-positive bacteria would be more dangerous, but in fact it's actually the gram-negative ones because of those lipopolysaccharides. These are considered toxins a lot of times when they enter our body, so these are the ones that actually tend to harm us quite a bit in terms of bacterial infections. Whereas gram-positive ones, these peptidoglycan layers, can actually dissolve if they're under the right conditions, like those inside your stomach, or if we increase the temperature in them, like in a fever. These lipopolysaccharides do not degrade as easily, and they can become toxic inside of our bodies. So that's something that makes the gram-negative much more dangerous to us. In the next activity in 316B, you're actually going to run a gram test on our bacteria that we found in our patients and see exactly whether or not we can identify what kind of bacteria it is based on the gram test. That's all for me. Let me know if I can help you out in your research on different bacterial morphologies, and stay tuned for that gram test on our patients.